going to get started. We're just going to start off today with our land acknowledgement. So I am joining you from the traditional territories of the Weewakai, Weewakam, and Queerka First Nations, where we're fortunate to live, work, and play. And I encourage everyone to acknowledge the lands that they are currently on and also have come from and been as well. And it's always cool to learn some of the history of those places as well. So if you have the time today, I encourage you to maybe just pick up a fun fact and learn something new about the area that you're in. We are today recording the meeting. So later on this week or next week, this will be available online for everyone to share widely. Or if you'd like to review whatever's going on in the meeting today, maybe you have some more questions that could be answered. It will be online on the CARTI website under our projects page as well. And finally, I'd like to just thank NSERC for some of the funding to be able to put this on. We've had some really cool participants this semester, and we're hoping to continue that as the year goes on. And we still have a couple of people trickling in here, so this is working out great. Today we're joined by Dr. Laura Dykman. She is over in Bamfield, or are you down in Victoria right now?
Currently joining in from Victoria. I move back and forth.
Nice. So both locations. Lauren's currently working for the Kelp Rescue Initiative, who she's going to present about today, which is awesome. Our talks so far to date have kind of been all over the place. So we've had some people from the East Coast of the US, had some people from Norway, from Alaska, and from Australia. And we've had, I think, one previous talk by Tom, who is here today. I saw him pop in. He's been our only kind of like BC presenter, and he was our first one. So this is kind of nice to kind of bring it into the full kind of circle, because he spoke about some of the green gravel work that he's been doing, and now we're going to hear some of the restoration work that is going on at the Kelp Rescue Initiative. So yeah, it's been a nice, fun series to this point, and we're going to try and continue this as we go. So without further ado, I'm going to hand over to Lauren, and we'll hear the Kelp Rescue presentation. So tune in, eat your lunch, and just have a good time. It's going to be a good time.
Great, thanks very much, Logan. I'm going to try to start sharing screen. So this might take a little bit. All right, now I've once again lost you. There we go. All right, does everyone over there see just the presentation slides and nothing else?
Yes, looks great.
Awesome, and you can all hear me all right?
Yep.
Great, so seems everything's set up. Thanks, Logan, for inviting me to the Seaweed Seminar Series. I'm really honored to have the chance to talk to all of you. I'm coming at you from Victoria, so I haven't had a chance to visit North Island College, but hopefully someday. It sounds really nice. And I am a member of Kelp Rescue Initiative, and I'm a postdoctoral fellow at the University of Victoria, so it's a joint fellowship. And today I'm going to talk to you about what we do at Kelp Rescue Initiative. sort of a broad overview. I'm obviously speaking for a large team, and I just moved here and started this work in February. So I'm just going to give an overview of what the team does, and then I'm going to go more into depth on two of my particular projects, which involve investigating the best methods to do kelp restoration by outplanting using green gravel, and sort of putting a genetics twist into that. So yeah, just a quick overview. I'm going to talk a bit about what Kelp Rescue Initiative does and a bit of the history, mission, and projects. And then the two projects I work on personally that I will be presenting involves investigating green gravel as a technique to restore kelp forests in British Columbia, and then doing some quick preliminary results on a study that's looking at the performance of different kelp populations under extreme ocean warming. A lot of you at this point are probably very aware of kelp forest ecosystems, but they're highly productive, diverse, and beautiful ecosystems that occur all along BC's coastline, all along the western coast of North America. And they perform a wide array of important services, including increasing fisheries yields. A lot of animals live in these habitats. They're really good primary producers, so they create food for other organisms and they produce oxygen. They also may have a role in sequestering carbon, and they're good at clearing up polluted waters. So there's a lot of services that we can get from these kelps, including important cultural and traditional purposes. And so just another example of how kelp forests are this important structuring habitat for a lot of species, and a beautiful habitat that humans recreate in and have food and livelihood from. And I just want to add this picture, and this isn't a BC kelp forest. This is a kelp forest from down in California, where I come from. So I grew up in kelp forests, and it was an important part of my childhood and upbringing to be able to go visit these systems and go snorkeling and go diving in them. So kelp forests are also important geographically because they cover 1/3 of the world's coastlines. And although there's a lot of research with climate change being centered on coral reefs, kelp forests are actually an area 5 times as big as coral reefs. So in this preliminary map, you see the extent of coral reef habitat located toward more of the equatorial areas of the world. And then looking at the range of various kelp species, these are more toward the cold water polar areas and temperate zones. So it's actually covering an area 5 times as large. Kelps used to be really large in extent and really abundant. This is an old newspaper from the Comox area, around where a lot of you are tuning in from. And this was from the late 60s, where it was actually at the point where they were calling kelp pesky, and they were using them as these fertilizer products. So kelp used to be all over the place and was used actually in industry. But things are not looking very good for kelp right now. And there are a number of reasons that kelps are more stressed now than they were even a few decades ago. And one of the interesting things about British Columbia is it actually has these pockets of really warm water that are not, they aren't really correlated with latitude. We often see along kelp's big range, it's much warmer in the south toward California and Mexico than it is toward British Columbia. But in areas along the British Columbia coast, especially in the straits, and for example, the Strait of Georgia, we're seeing temperatures that are as high as they are in Los Angeles. And this is like the farthest, that's the closest comparable temperature. So in this area, it's not latitudinally, temperature is not really latitudinally correlated. And kelp has been lost in the past years with ocean warming through a large range of British Columbia. So this map shows some preliminary data from some of my team members using mapping of kelp spatial extent. And the bar charts show the percent of loss and gain of those populations. So you can see that kelp have been lost in the past decades in a lot of areas, but this is really spatially patchy. If we zoom in, for example, on Bamfield, which is where I am going to be doing some of my research, this is a sort of at a strait and an inlet where there is a temperature gradient going from the open coast to the sort of more inner regions. And along this gradient, there have been larger larger kelp losses in the warmer waters. So the once again here, the blue on the map is a population that persisted through this heat wave that occurred in 2014 to 2016. And the red are kelps that have been lost during this really recent heat wave that killed off a lot of these kelps. And just to show this in another figure, this is actually showing the proportion of segments in this whole range that have been lost during that warming event. And we see that actually 43% of shoreline segments suffered kelp loss in very recent years. So warming is definitely a thing that's damaging these ecosystems, and we want to know how well they can recover from events like this. Another story you all might be familiar with was the loss of sea otters along the British Columbia coast due to the fur industry back in the 18th and 19th century, which led to the near extinction of sea otters. And the last ones were actually shot in this area in 1920s. And so they got completely removed from BC's coastlines. And now there have been some efforts to reintroduce the sea otters. And so in the 70s, a few sea otters were reintroduced, and they exist now in some places, but not in others. And sea otters are really important predators that actually eat some of the things that graze on kelps, which we'll talk about later. Another important predator that eats some of the things that graze on kelps is this sunflower star. And these were also decimated recently by this disease that you all might have heard of called sea star wasting. And this happened about 10 years ago, and the population still haven't recovered. So it's been sort of this double whammy. And the reason that these two predators are really important is there's an animal in the kelp forests that loves to eat kelp, and these are sea urchins. And sea urchins, by themselves are not a negative. We often talk about them in kelp as a really negative thing, but it's more just this imbalance where without these top predators, the sea urchins are booming in population and they get really hungry. And so they start overgrazing kelp forests beyond what the kelp forest can sustain. So this is an image from BC Shore Zone that shows an aerial footage of what used to be a kelp bed. And when you zoom in, you can actually see thick coverage of urchins all along the ground. And this is leading to what we can call a regime shift in this kelp forest ecosystem, where there's multiple stable states, and one state is a healthy, flourishing kelp forest. And then there's a shift into this state where it is very urchin dominated, and you see stubs of kelp and not as much of that structure. And it's really hard to shift from one of those regimes to another, especially when the kelp is climate stressed. So this leads to what Kelp Rescue is doing. We're trying to restore and increase the resilience of BC's kelp forest through a number of angles. And this is really a partnership between scientists, First Nations, and any actionists who want to conduct restoration. So we have a number of, and once again, I'm speaking for a large team here, but I want to sort of go through what some of the folks are working on. And we have a number of sites that we are working at right now, including the Broughton Archipelago. And this is a place where there's an initiative called the Broughton Aquaculture Transition Initiative, which is it's a collaboration of 3 First Nations where they're trying to remove fish farms and recover wild salmon. So we are helping with some baseline surveys of kelp in order to advance those stewardship goals. In Barkley Sound, this is that place that I said is a really good natural experiment where there's this big temperature gradient. So we are trying to conduct some experiments there to restore kelp forests. Hornby Island has been heavily temperature stressed. So there's also experiments going on there about populations that might be resilient to temperature stress. And then in Burrard Inlet, this is right off Vancouver, there's active restoration to try to bring back more urban kelp forests. So as I said, all of this is a big team collaborating between many universities and different researchers, like, for example, at Bamfield Marine Sciences Center. And we're all trying to tackle this problem from many directions, but also create actionable solutions. And through all of this, we are working on the traditional territory of First Nations, and we've actively partnered with a lot of First Nations in order to build knowledge and share knowledge, because Western science is one way to build knowledge, but there are many other systems of knowledge, and we can't fight these large-scale problems without everyone sharing and sort of building knowledge in a creative and different way. So the goal with our restoration is to sort of assist with these goals that communities might have, so that we can then turn the tools over to them once we've helped develop them. So just a quick overview of some of the projects that Kelp Rescue is doing. There's this large-scale blue carbon project that's trying to evaluate the amount of carbon storage in marine ecosystems in BC. And these are really using a lot of large-scale and small-scale ROV surveys, including satellite imagery and even just humans doing transects in the intertidal. And so a lot of this is looking at trying to make forecasting systems that can project kelp cover into 2050. We're also really interested in genetic diversity as a means to restore and make kelp forests more resilient. So we have great teams at UBC that are working on putting together high quality genome assemblies for these two big canopy forming kelp species in this area, which is giant kelp and bull kelp. We also have a team at UVic that is working on population genetics and looking over the range of British Columbia to see if there are population level differences or genes that might help with thermal tolerance. And then the final thing Kelp Rescue is working on is active restoration techniques using the kelp life cycle in order to culture baby kelps and put them out in the field. So as a quick overview, because you've probably talked about this at some point, kelp has a life cycle called alternation of generations, where it has this big stage that you can see, and that is asexually reproducing. And then it has a little microscopic stage that sexually reproduces to make these baby kelps that then grow back up in the field. So with Kelp Rescue Initiative, we are actually harvesting the spores off of these kelps. So you can see the reproductive tissue is one of these blades here. And we harvest that and bring it back to the lab, blend it up, and sort of get all those spores and start culturing these microscopic stages called the gametophytes. And then once we have those cultures, we can mix them together and they will reproduce and create young kelps. And now we do this in big aquaculture systems, and we spray these spores onto what's called, well, we call the product green gravel, but we're spraying them onto these gravels. And then kelp is going to be growing on these gravels in these culture systems. And you can see what these look like when they're really small. So these are just several millimeters in length. And these will get grown out until they're maybe a centimeter in length or a little more. And then they can be thrown out into the field. So this is an example of what outplanting one of these looks like. And this one's pretty large at the time of outplanting. But the idea with this restoration technique is it is affordable, scalable, and takes less worker effort than techniques that require scuba. So the thought with green gravel is you can just throw it off a boat and anyone can do it. You don't need specialized equipment. But it's a new technique that hasn't really been tested much, and it's just starting to be tested in BC. And so that is where my projects come in, is I was brought on to Kelp Rescue to do some of these field outplants and really demonstrate that this is a viable technique and figure out the best ways to do it. So I'm going to be presenting two of my projects today. One is in Barkley Sound, and this is by Bamfield Marine Sciences Center. And the point of this project is to figure out whether different populations or diversity levels will have better success on green gravel. So I said before, Barkley Sound is like a natural ocean warming experiment, where in this really small area, we have drastically different summer water temperatures. And this was also hit by a heat wave in 2014 to 2016, where sites farther up this channel were much more heavily impacted and saw a lot of kelp dieback. So the questions here, if local kelp extent here was reduced by 43%, we really want some kind of solution to bring this back. And studies in this area have shown that microclimate really structures kelp loss. Like they'll be lost on very small scales in really warm places, and then very nearby places they'll persist. So I wanted to see whether local populations that persisted through the heat wave are more heat tolerant. And in my study, we'll be looking at two sites, Dixon, which is this warm, thermally stressed site, and then Ed King, which is this cooler site. And I also want to mention that this is on the traditional territory of the Hawaiian First Nation, and we're hoping next summer to do more experiments in partnership with them to sort of advance what they, sort of the vision that they want for kelp health in this area. So this experiment specifically is looking at the number of parents that you are mixing, because in restoration, we want to know how many sort of source parents should we grab and then grow in order to outplant, and also looking at the temperature of the source site. So I said before, Dixon was really heat stressed during that heat wave, and Ed King was sort of a more normal temperature. So we made an experimental design where we have several replicates. These are the experimental replicates, and each of these squares is an underwater grid of 1 meter. So it's like a little plot that is 1 meter square where we plant out our green gravel. And the replicates have 4 different experimental treatments and a control. And the control is just to look if there's any natural kelp recruitment. So my colleague Clay Steele, who's the project coordinator for Kelp Rescue, gathered these spores and sort of grew them up in the kelp culture in different combinations of parents that we determined for the experiment. And that took place around January, December, January. And I wanted to show you what the 2 restoration sites look like, because they're really beautiful places to work, and I'm lucky enough to get to go out and scuba in these sites, particularly in the winter, which is real cold. So just some example of us doing the outplanting. So you might notice there is snow on this life preserver. So it was cold scuba diving. We were definitely in dry suits. And this is me and Clay holding our bins of gravel. And we had to keep them in these bins because since it's an experiment, we had to make sure that like the exact culture of gravel goes into the exact plot. So this was much more labor intensive than green gravel should be, but it's giving us really good scientific information. So a bit of a timeline. Clay gathered the sori, this is the reproductive structure, in December, and then they were cultured up and then crossed in order to make those sporophytes in early February. And then the sporophytes were confirmed in sort of early mid-February, and this is what I'm going to call day 0 for the purpose of studying the growth of the sporophyte. And then we outplanted them 24 days later and took measurements before we did so. And you can see this is an example of the plot where we marked the 4 corners with these brightly colored bags and put the little gravels out by hand. And then we kept some in the tanks and continued to measure them just so we could get growth rates in tank versus in field. And then we went back out into the field at 56 days old and we measured them. So that was just about a month ago. And our next measurement is next week. So we will finally get to see how they've been doing for the past month. So that's exciting. And just to show you some images of what this work looks like, this is me underwater holding a slate and a measure and a ruler, and you just sort of swim around and you count how many gravels have survived and measure a subset of the kelps. This is a video of what the green gravels look like after they've been out for 30 days in the field. So you can see on these white gravels, there's sort of kelp swaying around. And this is a pretty healthy looking plot. So the question is, how was survivorship and growth after 30 days in the field? And here I have the plot separated into the different combinations of numbers of parents. So this is like whether we crossed 2 parents together or mixed 10 parents all up in a culture. That's to look at genetic diversity. And then we have source population. So the warm population is Dixon and the cool population is Ed King. And what we can see from this figure is survivorship wasn't really, there wasn't any difference between these experimental treatments, and these weren't significant. But then I decided to break this up by the site we outplanted at them rather than the number of parents. And you can see there is a difference in survivorship at the two sites. So the kelps overall are doing better in the cooler site at King, at least 30 days in the field. These are all very early results of this experiment that is still running. We also analyzed length, and that had very similar patterns, but overall was fairly similar between the source populations and the number of parents, with the 10-parent culture having, at the warm site, having the lowest growth. Similarly, splitting it up between sites, we once again saw that outplant site made a significant difference, but the treatments didn't. So this is really indicating, at least in this early stage, that maybe genetic diversity and the source population isn't having as big of an effect as where we put the gravel. So this makes us start to think like where we outplant can have a very big impact on early growth and survivorship. But once again, all of these outcomes could change later on in the summer as the temperatures really start getting different. Since I said that outplant site made a big difference, I wanted to dig deeper into why that might be. So this plot, I'm simply showing all of the different plots that we were analyzing, and they are color-coded by the outplant site. And this is a plot showing survivorship. So all I need you to take away from this is that there was, in general, lower survivorship at Dixon, the warm site, than at Edking, the cool site. But there's also a lot of variation within that. There's also drastically different substrate composition at these two sites. So I started thinking maybe it's the percentage of sand or gravel or cobble that we outplanted these kelps over. So this plot is simply showing the breakdown of percent substrate composition in the plots where we outplanted our gravel. And this is in order of the percent sand. So I started thinking maybe the sandier plots wouldn't survive as well. And then the one pattern I did notice is most of the plots that had a lot of cobble were at Ed King, and most of the plots that had a lot of sand were at Dixon. So plot composition definitely could be one of the things driving this, because the sites show sort of different levels there. And so I did a regression, and this was actually significant, but it explained a low amount of the variance of the data, which means that sand might be, sand might be a poor place to outplant the gravels on, at least for early survivorship. But there's also a ton of other variation that's sort of hard to explain. And I'm going to briefly skip over this, but I drew a map of the site to see if there's any spatial correlation. And it seems at Dixon that the plots that were closer to the shoreline were having poorer survivorship. So the red is bad survivorship, the blue is good survivorship. And this could be because it's closer to the sandy area, or it could be because the island creates a shadow, and maybe the plots that were along the shore had worse light conditions. So there's like a lot of different things to dig into this with how to get the best results out of green gravel. So the main points from that study so far is after 3 weeks of being outplanted in the field, the green gravel overall showed 70% survivorship, and the kelps had reached on average a point of 52 millimeters. So they are surviving, they are growing, but there are differences in success depending on the outplant site, basically. So source population and number of parents didn't have a significant effect, which is, we had hypothesized they would make a difference, but they still might later in the summer. And then the outplant site did have a significant effect, but it could be due to differences in light and percent of sand. So I'm going to continue those within site analyses, and we have other experiments planned for next year to dig into that deeper. So I'm just really briefly going to touch on the other project that I'm helping out with, and this is at Hornby Island and Denman Island. So this is in collaboration with Hornby Island divers, and this is a diving group and a family-run business, and they've actually been trying to restore kelp to this area for many years, and so we're making it into a formal experiment. But the reason this area is really interesting to look at is it has experienced extreme warming in the past few years. So the Strait of Georgia, but around Hornby Island as well, has had a dramatic loss of canopy forming kelps. It has the warmest water temperatures north of Los Angeles. And in a study, they did find a warming trend over, a long time scale from 1950. So this area is warming on a climate change scale. And I just wanted to add in these aerial photos that my lab mate, Brian Timmer, is analyzing. But this is an old aerial photo from the 1970s that shows huge extents of kelp beds. This is right off Comox, actually. So this is historical data. And actually, the last kelps in this area died out in 2015. So just seeing the extent of change, where this is what the kelp beds used to be, and now they're completely gone from this area. So it creates really good motivation to try to find solutions to this large scale problem. And one of the rays of hope is that this is a kelp bed right off of Vancouver. So it's a really urban kelp ecosystem in a place that should be quite stressful. And it's still a very warm area, but it's holding on. And so we want to understand why and what is it that makes these kelp beds able to persist. So the experimental design for this study is to source kelp from various populations along this temperature gradient. So we have some that are in these cooler sites, and then we have a lot that are in these like areas of extreme warming that are still holding on. And we're going to outplant all of them in the same place off of Hornby Island right here in order to look at their performance in a thermally stressed area and sort of compare that. And for this experiment, we've outplanted them on seeded lines. So it's different than green gravel. It is similar to the techniques that aquaculturists use when they're farming kelp. So it's basically like growing the kelp spores on a little line and then twining it out on a bigger line and keeping that suspended above the seafloor. And this was from 2 weeks ago, and it's actually a really pretty setup. So this is bull kelp, I should mention. The past experiment was with giant kelp. So we're looking at 2 different kelp species here. So that's an example of what the array looks like. And basically all that we do is go out at various intervals and go along the line, grabbing kelps at random, and we'll measure them to take length metrics. And then we have a number of other measurements we're going to take at later points. But once again, this is still very preliminary results for this study. So I was just out there last week doing some diving and taking measurements. And what we have so far is looking at the length of their stipe over their number of days that they've been outplanted in the field. And we're starting to see some population level differences. For example, Browns Bay seems to be growing faster and taller, and it actually had reached the surface, so it's already canopying over there. And then there are some population level differences, but we're going to wait to see how that plays out throughout the summer. The other things we want to get from this kelp are growth and morphology. So we're going to be making marks on the blades and seeing how fast they grow. And this will get at these more fine scale differences between populations that might not be seen with just total length. And then finally, since this site is really close to the dock, it's actually a really easy site to work at. We're doing a few more pilot projects with green gravel to try to understand the role of grazers and the role of sand versus cobble as substrate in their survivorship. So once again, this is my coworker Clay, and you can see what this process is of outplanting the green gravel. But this is an experimental plot that's inside a cage that's meant to keep urchins out. And then the plot is divided. Half of it just has sand substrate, half of it has cobble substrate. And then we're putting an even number of green gravels in each half. And so this is basically, to understand how grazers play into it and what's the best substrate to put green gravel on. So just a very preliminary figure from that first outplanting time, or that first monitoring time. We see on the x-axis, it's whether or not the substrate had cobble, and then the colors are whether or not the plot was in an exposure that keeps out grazers. And honestly, there's a large difference between whether cobble was provided, and there wasn't as big a difference at this time point as to whether or not urchins were excluded. However, this was a time point several weeks after they were outplanted. And then we went back just last week, and actually the urchins had come in and done their work. So we no longer see any kelp in the unprotected cages, but we do see some really healthy growing bull kelp in especially the cobble side. So it seems like some of these differences are starting to manifest at later time points. So a summary of the talk today: Kelp Rescue Initiative is a group of scientists hoping to conduct cutting-edge science to develop effective restoration practices and enhance climate resilience in British Columbia. And then my project's Green Gravel. It's one potentially effective and efficient tool for restoration workers. It's not a solve-all solution, but it is one tool in the toolbox. And Kelp Rescue Initiative is trying to conduct experiments to figure out when and where this tool is useful. And then genetic diversity, which is the thread through all of our projects, is really key to resilience, because some genotypes perform better than others just due to variability. And we want to understand how to figure out where to source those and how to maybe use those for restoration. So I have a lot of folks that I would like to thank, and I have listed particularly here folks that have helped with my projects, but there's all of the funders of Kelp Rescue Initiative listed at the bottom, all of the First Nations whose territory we work in and who we are collaborating with, and my contact information is up at the top right if you want to send me an email.
Awesome. Thank you so much, Lauren. That's fantastic. That's an awesome presentation.
Thanks, everyone.
Yeah. I'll open the floor for questions. There's a few in the chat over here that maybe we'll start with, and I've got a couple more of my own to add in there. Looks like Caitlin's got a few here, so we'll start off with those.
Should I start sharing, or should I keep it up in case
You
people
can do either one. Is works for us.
I'll stop for now so we can all see each other bigger.
Fantastic. Alrighty, so we'll just start from the top here. Caitlin asks, are you coordinating with researchers studying if the timing of outplanting impacts recruitment, and were the shallower areas any warmer in your outplanting sites?
Yeah, those are great questions. So I do not know off the top of my head other researchers looking at the timing. So if you know folks, I would love to get in touch with them. That is definitely something that we want to look at in the future. We're actually just sort of getting our feet on the ground. So this year is some of our first outplanting experiments, and we have a ton of further questions we want to explore. And then as far as temperature and depth, yeah, generally shallow sites will be warmer, but it depends on the amount of mixing in the area versus stratification. So in the summer, we're definitely going to see more stratification where like the top layer is warmer than the bottom. So far at the times we've been going out, it has been very, very uniform and similar temperature in the water column, because we started in winter, and so it's just been cold at all the sites, and we're really going to see those differences in the summer. But one thing about the canopy forming kelps out here tend to like being around 20 feet deep, or like 9 to 11 meters, just because of light availability. So they really, even though it's like cooler down deeper, they tend to not want to grow there just because of that light trade-off.
Awesome. We have a couple more here. The next one is from Caitlin again, where the site is located on Hornby.
Yeah, so the site on Hornby, I can actually pull up Google Maps. It's a place called Maud Reef, but it's like right off. I don't know if you all know where Hornby Island Divers is. Yeah, so it's right by Ford's Cove Marina, but I will do a share screen real quick now that I have it up.
Now that you're a Microsoft Teams Pro.
Do you all see this?
Yeah, that's perfect.
Let me make the nice layers. So yeah, if you go to Hornby Island, this is about where the ferry gets in, and then you like drive around to Ford's Cove, and this is where the Zielinski family lives. It's a family-run business, and they've been trying to grow kelp in this area for like a decade because they are concerned about the loss of kelp. And so right off Maud Reef, they have this. It's basically the seeded line array that they put out there, and it's a really quick boat ride from their house, so it's pretty ideal for research.
Nice. Pat asks, have you noticed any European green crab in the areas where kelp loss is happening?
Oh, that's a great question. So not me personally, but I'm sure some people are looking at that.
Hmm.
Yeah, because like in the spots that we are out planting, I didn't really see them like directly there.
Awesome. Brent also asks, generally a bull kelp or giant kelp more resilient in terms of thermal stress and populations in BC?
Yeah, so that's interesting because generally giant kelp has a higher maximum temperature that they can withstand because they have their range tends to go farther south. So like they just seem to have a warmer optimum. But in British Columbia, bull kelp is like the dominant canopy forming species. There just isn't as much giant kelp, and they both are like are subject to heat stress. So I think that if you think on a population level, bull kelp might be more resilient just because there's more of it to start with. I think that if you lose like a small bit of giant kelp, it'll be gone. Whereas you can lose a lot more bull kelp and it will, just by sheer numbers, there will still be some left.
Super interesting. Awesome. Uh, Caitlin has another question, and she asks, how are you marking the kelp to study their morphology?
Yeah, great question. So for some of the population level growth that we're measuring, we aren't tracking individuals. We're just finding random ones each time. And then for the in like Hornby, once we start doing the morphological and growth measurements, we're going to be tying some soft, sort of any kind of soft like tape or material, maybe flagging tape around the base of the stipe so that we can, and then putting a little label on them. So that's sort of the nice thing about kelp is they're very easy to tag and track over time. And we're doing that at later points because like when they're really, really tiny, you can't really mark them. So now that they're bigger, we're going to start marking and following individuals.
Nice. Jen Clark asks, are there any lab-based experiments testing thermal tolerance of first generation crosses? Thermal tolerances of parents before first generation crosses.
Mhm. Not with this project currently. I think that would be a really exciting thing to look at. This is just these studies this summer are honestly a bit like pilot projects because I'm trying to make them as scientifically rigorous as possible, but it's the first thing we've done with this work out here. So that is definitely a question I want to look at because I think there's a lot of usefulness for lab studies at these early life stages. Honestly, like one of the exciting things for me about working with green gravel is a lot of people don't study kelp early life stages, and I think that's potentially a really important source of mortality is what's killing them when they're young. So yeah, looking at thermal tolerance of the parents and the offspring would be really cool.
Yeah, I actually had a really interesting conversation yesterday about like kelp microbiomes in like the lab and the juvenile phases. So there's some pretty interesting areas that you can explore in those younger phases that might have some longer term benefits, which is kind of cool.
Yeah,
Uh,
and I haven't been with our team. I haven't been the one involved in the aquaculturing or the sort of growing of the gametophytes either. So that's all stuff I want to learn more about. But at UBC, there's a scientist named Cami Good who has been doing that for that project, and then Clay Steele is the person in like our project that's worked on the Barkley Sound kelp.
yeah, yeah, that's awesome. That's super cool. Shanti has a couple of questions. The first one is, How far from the sites are you harvesting source tissue? How do you determine site appropriacy? And finally, is there any concern of interbreeding with wild kelp?
Into bringing what? What was the last question?
Interbreeding with wild kelp.
Oh yeah. So the first experiment in Barkley Sound, we are it's called a reciprocal transplant experiment. So we're sourcing the kelp from the place where we're putting it out. We're just sort of swapping two areas, but they're within like a few kilometers of each other. So it's not we're not bringing them a far distance, and that's sort of why we're trying this in an area that has like really steep temperature gradients over a small space. Is then you don't have to really worry about the ethics of transporting kelp really long distances, because like feasibly these are areas that are oceanographically connected anyway. And so yeah, the crossing of parents, they're all parents that could have crossed in their ecosystems anyway, but we are just having complete control over who crosses with who, which gives us really good info on who the we can like genetically track back who the parents are. And then yeah, for the one in the Strait of Georgia, we definitely sourced kelp from much farther away. So that does have, some ethical issues, but we're outplanting it in a site where the kelp has been completely destroyed. So there's like no chance that they'll be crossing with wild populations. And also the summer temperatures tend to kill them anyway by like July. So they haven't actually succeeded in returning a like persistent population to that area. But yeah, I think that's a huge ethical consideration in restoration, where it's how far away should you be taking your source populations from?
Yeah, a lot of the work that has been going on with us, we've been kind of following the regulation or suggestions of the 50-kilometer radius and using that as our main guideline.
Yeah, I think it's going to be
Yeah.
a while yet before we start thinking about bringing, California kelps up to BC. I think that's something people need to have a lot of caution before thinking about.
Yeah, yeah. Larry asks, where in Barkley Sound is the experiment? I think there was a little map at the start,
Yeah.
right?
I should open up Google Maps again.
And while you're doing that, Mark Donahue has a quick question. How many pieces of gravel can you produce in a batch or tray for the green gravel?
Yeah, I mean, it honestly depends how big your tray is. So I don't think there's really a limit as long as you have good water flow in your tray. Trying to remember off the top of my head the dimensions of our trays, but they were maybe like this big. I can't remember the numbers. And we put, I'd say like 40 to 60 gravels in there. It also depends on the size of your gravel, because a lot of these studies started using pea gravel, the really tiny ones, and we were trying to use like 2-inch gravel to sort of make them less likely to drift away or easier to count out in the field.
Yeah.
But yeah, lots of them.
Yeah,
And
one of the things that
I
we've
can
run
share the
into.
map.
Oh yeah, go for it. One of the things that we've run into is we can't actually collect gravel from like the beaches or locally for some of our projects. We're having to like purchase it. We have
Yeah,
to use it.
we purchased all of our gravel, which
Yeah.
costs. So that's one of the places where this method is costly, is if you have to purchase rather than harvesting off a beach. So anyway, this is Barkley Sound. It's this whole area. And then if you zoom in, there's Bamfield and the Bamfield Inlet. And so we're leaving on our boats out of here. And then for the Ed King site, we're transiting with the boats through here and around to here. So it's sort of at one of these outer islands on the Deer Group. And then the site at Dixon is just a little up the coast from Bamfield. Out this is Dixon Island, and there's even by a place called Kelp Bay. There is a remaining kelp forest in this area. So with this study, we are outplanting in places where kelp still exists. It still has been able to survive.
Nice.
Can I
Sarah
say
has. Oh, go for it.
Yeah, I'm from Bamfield, so yeah, Kelp Bay. It used to run from Dixon Island. There'd be kelp all the way up to the top end of Rockafell Bay back when I grew up. So yeah, it's interesting that you're doing that. Any success on natural after you put some in the area? Is it naturally recruiting?
We can't say yet because we just outplanted a few months ago, and so it would have to get up to reproductive size. And we like if you're asking about like the natural populations recruiting, we might be able to get more info on that just next week when we do our next sampling, because we might start seeing them like big enough to notice.
Okay, thank you.
Yeah. But there definitely is still a surviving kelp bed, and something interesting relevant to your area is that kelp has started to grow prostrate, where it's actually not reaching the surface, but it's growing along the bottom. And no one knows why this is, and it actually makes it hard to estimate kelp cover from the surface, but it could be an adaptive thing where they're trying to get lower down and not reach up to the really warm temperatures. But once again, scientists don't really know how they're doing this or why they're doing it. So at Dixon, there is like a good prostrate kelp bed, even though we don't see it up at the surface.
Cool. Sarah has a question here. Have you thought about sourcing the kelp from oyster river sites for hornbeam outplants?
Yeah, I think one of our sites actually is Oyster River, so that is a site of interest for the future. And I'm not as familiar with the area because I'm just joining the project, but that is one of the existing kelp beds that's nearby. Correct? Like one of the only ones.
Yeah, it's pretty close by to that one.
Yeah, and I saw Bill Heath just added or joined the groups, but I think that bed is really interesting because it's at the mouth of fresh water and could be an interesting.
Yeah, so the folks who've been working on that project a while, Rob and Amanda Zielinski, they looking at Oyster River, and they're extremely interested in like studying what oceanographically is like causing the kelp to be resilient. So
Totally.
yeah.
Thanks.
Jen Clark has another question of whether you've made any considerations on the type of substrate, granite limestone and roughness.
The ones we were using, I'm not sure the exact type of rock. It could have been granite. I don't think they do well on limestone. It might alter the pH of the water in culture, but I think ours might be granite. They're just like very white, smooth rocks. And we just don't have the info yet of having tested a lot of different types of gravel. So that is definitely also a future direction. But there's other groups that have worked on various different things. So I think we all just have to get together and sort of share that knowledge because it's not always published.
Yeah, and I mean, I'm gonna keep on the green gravel questions here, but I guess that's part of it, like sharing the knowledge there. My question was if you considered the size of your green gravel in terms of like the kelp buoyancy and its like drag properties when it's in current as it grows, and in like a relationship with the holdfast and how fast that grows to that like grow to the substrate that's below it.
Yeah, that's actually one of our main future directions. So there's a flume at Bamfield, it's like basically a water racetrack where you can make water run at any speed you want. So we're thinking that might be a good way to do controlled experiments on the drag of different size kelps and the size of the gravel. So for people's information, the idea with green gravel is that the holdfast will grow off the gravel onto a larger piece of rock on the substrate, but we don't yet know how fast the holdfast grows. So it basically has to grow fast enough that it can latch onto something else before the kelp becomes buoyant and drifts away. So that's definitely like, I think it can be done. It's definitely a trade-off of different gravel size and growth rate. And that's just an experiment that has to be done. And I'm like really interested to look into that.
Yeah, that's one of the things that I'm super interested in as well, because I've seen in Australia they've used really big pieces of rock for their green gravel, but then it's like a trade-off of like how much space you have to actually create that green gravel versus how many outplanting objects you have, and whether or not you can outplant lots of little ones and it has like a great effect, or you need the bigger ones to have that hold fast versus like the buoyancy of the kelp. I think there was a really interesting paper that was from UBC that looked at like the drag properties of bull kelp, and as it grew more blades, it had less drag in relationship to its size, which I thought was super cool. It was a really cool project.
There's a lot of like really in the weeds hydrodynamic stuff we could do, which would be super cool.
Yeah, super interesting. And on the green gravel, more questions for that. I wondered if you've considered the number of kelp per green gravel, and whether or not if you have too many on one, you have kind of a crowding effect where you don't get the growth. Because what a few people are seeing in certain cultivated kelp projects and even on some farms is you get like this crowding of holdfast and then just a few smaller kelps. Mostly this for like some sugar kelp. You get some smaller kelps growing, and then alongside it'll be less crowded, and you get the really big kelps growing. So I wondered if you'd seen any effects like that with your green gravel.
Yeah, so when we went out before, and at that point they'd only been outplanted 3 weeks, so unfortunately I just have preliminary results. But at that stage, they looked like there were a lot of kelps per gravel, and they all seemed to be still doing pretty well. So I think competition hadn't become a huge problem yet. We definitely expect at later stages, like 1 or 2 kelps will dominate and win out. But whether that competition sort of hinders their growth isn't really known. Like I think generally there will be like one successful kelp and it might do fine. But yeah, it probably isn't doing as fine as it would if it had the gravel all to itself. But I think there are likely, at this stage, it seems there are other factors other than crowding on the gravels that are probably doing a larger role in killing off kelps on the gravel. Like even things that aren't grazers, like sea stars crawl over them, or things they get flipped over by the current, or like a fish flips them over. So there's like a lot of things that could kill a baby kelp at early stages. So I think they do self-thin quite a bit. And also the act of throwing them off the boat in the first place, like we do lose some, but we honestly consider it sort of good because then they're competing less with each other.
Yeah. Oh, that's awesome. I'm really interested to see like what your some summer survivorship is going to be like, and especially comparing between having like the like cobble or gravel underneath and then the sand, as well as like the temperature and things like you mentioned the water stratification.
Yeah.
What we find when we're up in some of the inlets here is that we get like really high depths of freshwater lenses on the tops of these areas, and I'm really interested to see how those interact with your outplanted kelps. But there are a couple more questions in the chat here, if you've got some time. There's one from Melinda asking about if there are notifications for the research projects, just to make sure that there are no overlaps with current or proposed aquaculture.
Yeah, so I am aware near our near the Barkley Sound site there is shellfish aquaculture that's adjacent to ours. I'm not aware of other types of aquaculture. I know that Nova Harvest, the oyster place, is also working on some kelp culture, and I'm not sure where they're putting that out. Hi. So yeah, if you have any information, I know that we do go through BC regulatory bodies whenever we propose and do a project. So yeah.
Perfect. Yeah, I just wanted to flag. It looked like one of the sites was where we're seeing a lot of interest for aquaculture for aquatic plants. So I'm like, ooh, if we don't know about a project, we're getting applications that we may be approving if we don't know a research project. So that was just an information sharing to make sure that we're well aware.
Is it the ones at near Hornby or the ones near Barkley Sound?
The Maud Island caught my attention, as well as the one in the Deer Island Broken Group, I think it was out there.
Yeah, okay.
The West Coast.
if there's anything we should be aware of, so we're not, you know, infringing on other people's grounds, let us know.
Yep, I think we can share the website just to make sure you know where the proposals and applications are, so you're most up to date with everything.
Yeah, and we also know that you know, once our kelp is out there, it belongs to the Crown, so it can be harvested. We've put our buoys out to be like experiment in progress between these places, but if the kelp starts canopying, it's like anything's fair game. You know, that's all part of the experiment. See if humans start to use it. Yeah.
And Leanne has a suggestion for sharing the projects on the Kelp Forest Alliance or Green Gravel Action Group, which you guys might be part of.
Mm-hmm. Yeah, I've joined a meeting of the Green Gravel Action Group, so we should be up there. I don't know if there's like formally a page on these projects, but yeah, we're definitely collaborating with them pretty frequently. I haven't interacted with Kelp Forest Alliance site yet, but I've also like been working with the folks on that group. So
yeah. Awesome. Cool. It doesn't look like there's any more questions coming through in the chat box, but if anyone has a quick question, feel free to just use the raise your hand and we can answer it. But other than after all those questions, I think we might start wrapping things up. So thank you again, Lauren, for presenting on this. It's awesome to bring things back to BC. And for everyone else, the recording will be available on the website as we have done with the previous presenters. So if you would like to view it or share it with anyone, please feel free to pop on there in a couple of weeks when it's up there. But one of the last things I just wanted to mention is I think we're going to be taking a quick break for the summer. So we will have more Seaweed Series presenters and exciting projects coming up. Hopefully the first one will be back in either August or September, and then we'll have some more people throughout the year, and we'll keep it going over the winter area. But yeah, thank you guys all for coming and supporting this, and hopefully learning something over the past few months. It's been super fun, and yeah, we'll see you guys all in the fall.
great. Thanks everyone for attending.
Cool. Thanks again, Lauren. That was awesome. Really, really
Thanks.
cool stuff you guys are doing.